So how do we identify if the bacteria has taken up our recombinant plasmid? So let me just give you a quick overview. So this is our desired gene, insulin gene. We have extracted it. Then we're going to join it with the plasmid using DNA ligase. And then we have our recombinant plasmid. But we're not sure if the bacteria will take this recombinant plasmid. Most of them will not, and only a very few will. But how do you know? And that is the part where the examiner will ask you the gene markers. You don't need to memorize the exact steps, but this is what you need to know and write down as a mark scheme answer. So I'm going to show you the key words you need to write down for one of the gene marker, antibiotic resistance. So let's make it very simple for everyone how to do this in the exam. Question would be, describe how you will identify if the bacteria has taken up our desired gene, meaning the plasmid which contains the desired gene, recombinant plasmid. The first thing you would say, insert the antibiotic resistance gene into the recombinant plasmid. So we're going to initially, at the beginning, we're going to make sure our antibiotic resistance gene is in here, in the plasmid, recombinant plasmid. And then number two, grow the bacteria with the recombinant plasmid. We're going to grow the bacteria with this recombinant plasmid. What does it contain extra? Our antibiotic resistance gene. Number three, make sure you say do replica plating. Very important. And replica plating essentially is when you transfer some of the bacteria from the agar plate, from that medium, into another medium that contains antibiotic. So just you have to mention the word do replica plating, key exam word. And then what you will see in the next new medium that you have transferred to, that the colonies, bacterial colonies, some of them will die, will be killed, and others won't. So the colonies that was not killed means that these bacteria contain the antibiotic resistance gene, and if they contain that, which means that they had the desired gene. That's the most simplified way, and it's most likely you can get away with that in the exam if you mention these important points. Remember, it's only, you don't need to know the specific way, just need to have an idea how antibiotic resistance gene can be used as a concept to identify the bacteria that has taken up the desired gene.